Hello, welcome to the third part of our lecture about organelles. This one is bacteria-like organelles. We're not going to cover these that in depth just because these will be something that we cover in the next couple chapters, but I just want to introduce them, that way you guys know them, and hopefully you already do from other classes. So bacteria-like organelles, they're the organelles in the cell that release and store energy. Now there's really two main types, and that's mitochondria, which releases energy and creates energy for the cell, and chloroplasts, which takes in energy and stores energy in plant cells. There's also with the chloroplasts in that family called plastids, and there's a couple other plastids in plant cells that we'll talk about in this presentation. So real quick brief overview of mitochondria. Mitochondria have their own DNA. You actually get the DNA from your mitochondria from your mother. So when we do DNA tests of mitochondria, it always follows the maternal line. So you have the same mitochondria DNA as your mother, as your grandmother, and so on and so forth. And that's how we can also trace back lineages like that. The mitochondria are bound by a double membrane. This is a main principle of the endosymbiotic theory, or endosymbiosis theory, however you want to do it. But it also brings a huge role when we're doing cellular respiration, when we talk about in a couple weeks here. Now, when we do cellular respiration, we actually break down either glucose or fatty acids depending on what we actually have as a energy source. And breaking these down, we actually get ATP out of this. As we learned before, ATP is like the money of the cell, and it's the energy of the cell. So if we need to do a chemical reaction and it takes 10 ATP, then we need to create 10 ATP to do it, and we do it usually by cellular respiration. Chloroplasts are derived from photosynthetic bacteria. That's part of the endosymbiosis theory. They take in energy from the sun, they convert that energy into sugars, and they do this by it seems like a very complex way to do it, but it's actually quite simple and it's pretty easy to follow, and you guys learn about it in biology, and we'll learn a little bit more about it in this class. But basically it just takes the sunlight and you get C6H12O6 out of it. I brought up in the beginning about other plastids, and these are plastids we don't really talk about a lot just as a biological community, but they do play a major role, and we actually kind of lie to some young kids about their role, especially with the chloroplasts and the chromoplasts. Now, the chromoplasts contain pigments that result in the yellow, orange, or red color. We say to the young kids that the chlorophyll breaks down and you see the xanthophyll and stuff like that, but actually a lot of these things are in the chromoplasts, not the chloroplasts. So that's what is responsible for the leaves in the autumn, for fruits, carrots, especially carrots like the carotenoids, which is the color orange. Leucioplasts are a little bit different. You find these in potato plants. They're colorless. They're really hard to see underneath a microscope, so we actually didn't know about them until about the 1970s. But they synthesize and store starches and oils. So these turn sugars and complex sugars into the longer lipid chain, so the plant can actually store them in the winter. So it's a major part of cells, especially in the roots of cells, and when we look at, like I said, the cell, you won't be able to see these, but especially in the root cells, they're there. Now the rest of the slides are really just an overview of everything about cells. It goes briefly through each organelle, their description and the function. This is not an exhaustive list. There are some organelles we talked about that aren't on this list. This is just a list from a book that I found. And the last slide is a look at the differences between eukaryotes and prokaryotes, and within the eukaryotes what the differences are between plants and animals. So I'm going to stop talking, but I will let you guys, I'll stop in each slide for about 3 seconds, and then I will stop the presentation. Make sure you stop each slide, you look at it. If you need to take any notes down, you take some notes down. But that is really all. I will see you in class. You completed the 3 different presentations. Good work. And I will see you guys in a couple days. Thank you.